The other, I think, big thing in this is what are we doing about KAITLIN? Does someone want to talk about KAITLIN? You know, those results? Mark, do you have that?
Yeah, I've got it. I mean, it was a substitution of T-DM1 for paclitaxel and 2 antibodies. It was T-DM1 plus Pertuzumab. And the trial did not meet its primary endpoint. So chemo HP remains the standard of care. Moreover, there were more dropouts in the T-DM1 arm, although the quality of life was a little bit better during the T-DM1 sequence in the trial. But the bottom line is it's a negative study. You know, maybe only for selected patients who can't tolerate paclitaxel and 2 antibodies would it be a consideration. But for the vast majority of patients, it's an interesting trial. Didn't meet its primary endpoint. It's not superior, but it probably is. It looks to be, you know, highly similar, if you will, to the 2 antibody chemo control. So there'd be no barrier to using it in terms of efficacy, but really no advantage.
They made a big deal, though, about how it was not non-inferior. You know, so they're trying to tell you don't use it.
Don't use it.
On the other hand, you're going to have a woman who comes in who doesn't want to lose her hair. You know, what about then?
Right.
Now that's probably the only time I think about using this regimen. What do you think, guys? Virginia, what do you think?
Oh, you're still using AC, so.
Yeah, I forgot. That's true. You're going to lose your hair, so that doesn't count.
I think
this
is not the only trial that has shown kind of similar results with T-DM1 in the neoadjuvant setting compared to a taxane-based regimen. And then you look at the cost and you realize it's probably not worth it.
Especially now we have biosimilars, so that's a good point. You know, I guess the question, though, is, you know, so why? Can anybody explain why that would be? I mean, I think it's a really interesting scientific question. You know, shouldn't it be better? It works as salvage therapy in the metastatic setting, but it's not better in the first-line, frontline setting. I don't know.
Well, I mean, that was shown last year by multiple trials that T-DM1 cannot accommodate HER2 heterogeneity. And in the Dana-Farber study, about 10% of their patients were heterogeneous, and they had a lower PCR rate to T-DM1. The same was true in the Christine neoadjuvant trial, also about the same frequency. About 10% or so, you know, just didn't respond from the get-go during the T-DM1 neoadjuvant treatment. And I-SPY2 also showed similar results, that T-DM1 as a single agent, since it lacks a bystander effect, DM1 is not soluble. It can't diffuse into adjacent tumor cells after dead cells are lysed. So that's a big handicap of T-DM1.
Yeah, no, I think that's a good, that point is an excellent one. I think the fact that you need functional HER2 expression to have it. And if you don't, or not enough amplification, it's not going to work. I think that's an important clinical pearl. I mean, you know, you're ready to reach for the T-DM1, you know, part of this, you know, as you know from San Antonio, you know, the whole, I think it's ATTEMPT was the name of the trial. Is that right? You compare T-DM1 to TH. You're ready to reach for it, but the woman has heterogeneous disease. That's a very, I never would have thought of that. That's a really, again, you want to comment on that? That's a really good point, I think.
I mean, the other issue, Adam, is dose. You know, T-DM1 maintains all of the attributes of trastuzumab, the naked antibody, and it's insofar as it has signal perturbation in terms of HER2-HER3 association. It engages activating Fc gamma receptors on NK cells. So if any of those are also important to the mechanism of trastuzumab with chemo, you probably lose some of that with T-DM1 because of the lower dose, much lower dose, almost half, and the half-life is far shorter with T-DM1 compared to free antibody. So that's another mechanism of potential failure.
Yeah.
I didn't realize that. I thought, so the dose is 3.6 per kg, right?
Yep.
We're every 3, where the
half-life is far shorter. The half-life is less than a week. The trastuzumab half-life is 3 to 4 weeks.
Yeah, you know something? I never thought of that either. These are very good points. All right,
here's a question. When you look at the presentation that they did, and we'll talk about Catherine, but it seems that they presented data at ESMO Breast about the patients that post-neoadjuvantly were HER2 negative, and these patients did get benefit from T-DM1. So how is that explained?
Yeah, that's a great, let's answer it. I don't know the answer. That's a great question. Mark, what do you think?
Well, I don't think that in the case of T-DM1, the question of HER2 low has been critically addressed. There's been a small study, I believe, but in that study, HER2 low included 1+ and 2+. The current IHC antibodies that are commercially available for HER2 testing were not designed to discriminate the nuances below 3+.
Yeah.
And so I can envision, especially in the case of micrometastasis, there may be some cells that have just enough HER2. All you need to get is internalization, and then DM1 can nicely kill the tumor cells. But you have to have enough. And these heterogeneous cases seem to be admixtures of truly HER2 negative cells mixed with HER2 positives. Those are the ones in which TDM1 seems to fail, at least in the neoadjuvant setting.
These are all good points. Excellent points.